moreover is in below ground symptoms, we can see the rotting of the roots, the rotting of roots and rootlets. Also, we can see, and moreover, sometimes we can see the development of adventitious roots, especially at the collar region, is also we can see in this problem. And with respect to diagnosis of disease, we can take the infected plant and we have to dissect the stem at uh, you can go for cut the plant across the stem. You can see you can kept this the infected step inside the water, the clear water. You have to see this if you find any kind of flowing of the whitish fluid or oozing if it is coming from that infected surface, then we can identify this bacterial disease problem in tomato. And also we can see the yellowing of the stem, the hallowing, because of rotting of the internal the tissues, that especially the pith portion of the stem, that results in hallowing of the stem, is also one of the typical symptoms of the disease. And moreover, the disease is very problematic wherever if it see the temperature exceeds 32 degrees Celsius and more than that. So only under such situations we can see this bacterial disease. So this is the picture. We can see this the symptom of this bacterial disease. So the sudden wilting. See, we can see still the leaves, which is greenish in color. The plants are expressing this wilting, that is the drooping like symptoms of the leaves. It is very common at the field stage, and we can see the development of adventitious roots. So we can see that at the color portion where we can see the development of adventitious roots is also one of the symptoms of the disease. And we can see you can cut the stem um, transversely, and you kept this infected stem inside the clear water. We can see the flow of whitish colored the fluid. So based on this ooze test, we can easily get this diagnose this problem. Then, with respect to the etiology of this bacterial disease in tomato, no, the Ralstonia solanacearum, it is a gram-negative bacteria. Most of the disease causing the bacteria in plants, they are all belong to this gram-negative, and we can see the polar flagella. That means only one side of this bacterial cells. You can see this presence of flagella, which are many number. You can see this is also one of the morphological characteristic of this Ralstonia. Then it is a aerobic bacteria, so it requires oxygen for its growth and multiplication. And one of the important thing is this is having the wide host, nearly about 200 plants. This pathogen going to cause the disease, which is belong to the more than 50 families. Uh, this is it is um, Polyphagus pathogen. And with respect to the primary source of inoculum, the primary where exactly this pathogen is going to survive in the plants. So it is mainly going to survive in the infected plant debris nearly about 2 years, and it is a seed-borne in nature. So seed-borne in nature of this bacterial disease, you can see in this crop. And with respect to the secondary source of inoculum, that means where exactly which is the inoculum which is going to cause the further infection. So it is usually the bacterial cells. That means it doesn't produce any kind of spores. So you can see it in fungal pathogens. We can see the new different colored, different shape. The conidia we can see, spores we can see. But in case of this bacteria, whatever it may be, whichever the bacteria, we don't see any kind of the spores. But here, the bacterial cells. So bacterial cells consider the source of inoculum, and mode of dissemination. This mode of dissemination is mainly by through irrigation water. So if you irrigate the field which is infected with the this pathogen, along with the water, it can be able to move from one place to the another place. So that's why whenever if we go for managing this disease, you should take care of this irrigation practice. Also, one of the important the strategy for disease management. So, with respect to the epidemiology, this I already explained about this. It requires more than 32 degrees Celsius of temperature with